I'm going to turn the volume down. And it's still somehow too loud. Jesus. This wouldn't be a fucking problem. Lo and behold, it is. Okay. Back in. Back on. Jesus Christ. Itchy. I have a new fit. Just for today, since we're kind of going ham with the teacher vibes. Well, not teacher, it is TA. It is specifically TA. There is no implication about high school. There is no implication that I am like an adult with power. I am a teacher's assistant. That is it. Also because I'm not really going for like the vibe, but I thought it was fucking funny. And that's what matters. Phone, get out of the way. Exactly. Put your phones away. Sit the fuck down. Lock in. Where is my water? Sweet Jesus. This wouldn't be that fucking difficult. Okay. And I'm gonna have the mic nice and close to my face so that way you can actually hear me, hopefully. Let's make sure that everything's working. Yes. Do tell me if the music is too loud, or if my mic is too loud, or if there's any of that shit going on. Feel free to let me know on that front. Hi. Welcome to class. You're late. Sit down. Okay, good. I'm glad the sound levels are good. You're late. Sit down. Don't interrupt the rest of the class. I say the rest of the classes. I think that's just you. We are learning today. Specifically, this is probably one of my favorite classes right now. So I'm doing a couple different classes. I'm doing neurobiology, intro calc, intro chem 2, and then this one is the birth and death of a cell is what it's called. So the first half of it was cell birth, which I'm not studying today because I've already written the exam for that. The second half is on cell death, which is what we're going to be studying today. It is surprisingly short. There's only 28 pages out of my notes, which is really wonderful, actually. So necrosis is where we're going to start. So cell death is not a bad thing. It never has. It's not a bad thing across the board. Cell death does in fact have a place. Like there is a healthy method, and as you age, your cells will eventually just die because they can't replicate. They will either die because they can't replicate, or they just go into something called cell senescence, which basically just means that they're no longer dividing. And most cells do have this limited replicative capacity. You've probably heard about this in the context of neurons. Like, you have, like, as many neurons as you're ever gonna have, is, like, usually what people say, which, for the record, isn't 100% true. But we'll get into that later. So, generally speaking, the healthy method of cell senescence is telomere shortening. So, telomeres are the little caps on the end of your chromosomes, and as your DNA replicates, parts of your chromosomes will kind of like break off because replication isn't perfect and errors happen all the time. This is also why the Y chromosome is dying, but I'll talk about that in greater detail after this. So, telomerase is not expressed in the vast majority of your cells, and it's generally not expressed ubiquitously in humans. There are cases where it's expressed pretty widely. You've probably seen like those little teeny tiny little microbial pigs. They're not pigs, but they look like pigs. Like naked mole rat pig things. Those have telomerase. So they are effectively immortal. Similar thing with jellyfish, but jellyfish go back to being children. So the 3' end doesn't quite get made to the end and is replicated. This is every single time that you replicate your DNA, this happens. So over time, your telomerase caps just kind of disappear, which is fine and normal. Stem cells, in contrast, do have telomerase because, you know, stem cells can theoretically become anything. And it also can be manually expressed using certain treatments. You do see like a younger phenotype in older mice when you treat them with this, but, it's not really great to try and do this in humans, largely because we don't really know about what kind of effects this is going to have. Like, you see a young phenotype, but what is underneath that? Because the phenotype doesn't necessarily always represent the entire genotype, so we can't just kind of go, here, have some telomerase and you'll live forever. That's not, unfortunately, how it works. In the case of an unhealthy or bad method, is mostly DNA damage. You're gonna see this a lot, Ross. It's reactive oxygen species. Hello, allotments. Hello, we're learning about cells. You're late, sit down. So reactive oxygen species usually pop up out of your mitochondria because in the electron transport chain, which is where ATP or our energy source comes from, there's oxygen that kind of gets transferred all the way through because oxygen is nice and electronegative and pops out electrons all you want. But you can end up with reactive oxygen species, which are, in the term, reactive, and will damage the fuck out of the area around you. So, not ideal. We do have ways to clear them out. And the problem is when it's on the nuclear DNA. If it's on just kind of like some other shit that's clicking around, it's more repairable, but DNA being the blueprint, if you break the blueprint, you kind of break the rest of it, right? And in the cell cycle, there are a ton of checkpoints, which are very, very strictly regulated to check for this damage. So checkpoint is engaged and stops progression because it kind of goes, hey, don't do that. So the cells will still be alive, but they won't usually divide. If they prove to be a problem, they'll be marked for degradation or being consumed in autophagy. I thought this was autophagy for the longest time. It is autophagy, that is how it is pronounced apparently. Refers to like self-eating is the term, I think it's ancient Greek breakdown. Auto is self, phagy is eat. It's degradation and recycling of the little bits inside of your cells. It starts with the goal of cell survival, because autophagy doesn't necessarily mean that you eat the whole cell. It can just be kind of go, hey, this part is fucked up. So you can theoretically just consume just that part, which is the goal generally is cell survival, because making new cells is like takes resources. So if you can just kind of engulf the damaged or unused organelles, great! Unfortunately, sometimes you do need to consume the whole thing, which is usually where you see macroautophagy, which engulfs the cytosol, which is the inside cell fluid. Sorry, I'm so EB. Okay. Autophagy is required for survival. Certain diseases like ALS arise when it just kind of doesn't occur. Hold on really quick. I gotta define what ALS is, because I define it later. Amyotrophic lateral sclerosis, the nervous system disease that affects nerve cells in the spinal cord, causes loss of muscle control, gets worse over time. Yeah, I would end up pretty much knocking out most of your movement. Because you need to get rid of the dead stuff or else the dead stuff clogs up really bad. So, we will come to this later. I'm not going to go into it in depth, but we will come to that again later. But in non-selective autophagy, it just kind of goes, yoink, and just consumes the whole damn thing. But in selective autophagy, which is much more common from what we've been studying, is it's marked for autophagy, and then it ends up being enveloped by this little envelope thing here, fuses, gets spit out into a vacuole, and gets degraded because inside the vacuole there are normally really strong, low pH enzymes. The autophagosome is kind of that little like multi-membrane thing here I was telling you about. It has two membranes in it. The reason why it has two membranes in it is so you can see it in this picture right here, and I'm going to try and zoom in a little bit to make it easier to see. Okay, so there's two membranes, and there's two membranes in this one here. There's the outer membrane, the inner membrane, yada yada. So the outer membrane will fuse with the membrane of the vacuole, and then you still have the inner membrane left over, so that way it can be degraded. The reason that way it can just kind of like fuse into it and not like break through or anything like that. This is very common with most methods of transport. Okay, so LC32 is what we're going to refer to that one as. It's a marker for autophagosomes. You also see ubiquitin coming up very often. Ubiquitin is generally just a target for please degrade this, but ubiquitin is usually the tag for please degrade this in the proteasome, which is basically a big tube of proteins that it goes through and it breaks down and dies. But it can go either way. Now there's two different types of like lysine markers here in ubiquitin. So 63, lysine 63, which is going to be lysine at the 63rd amino acid position, is associated with autophagy, and then at 48 it's associated with proteasomal degradation. So you can have the same overall protein structure technically, but that tiny little bit will mark it for a completely different sulfate. You do have LC1 as well, but it's in the cytosol instead of being on the actual autophagosome itself. Kind of similar to ubiquitin pathway, which I may have talked about before if I ever taught this course. I don't remember if I did. So ubiquitin marks for degradation in the proteasome, and LC31 marks for degradation through autophagy. It's kind of the same process, just with different things. And I imagine that the actual, like, nuts and bolts of the process are different, but overall, this is what you sort of see. Now, RAP will come up again, because there's a bunch of them, but we're going to talk about that later. So RAP induces autophagy. You can stick a green fluorescent protein, or GFP, onto it. It is something that will fluoresce. If you like attach that on there and it doesn't get cleaved off, you can then have a live cell and stick it under a fluorescent microscope, and you can watch as it moves, see where it localizes to, and then when it breaks down, and then you can determine like what's happening to this protein over time. What is it doing? Yada yada. So there's a drug called baflomycin. It blocks lysosomal fusion degradation, which is this shit right here, meaning the autophagosome won't actually fuse and you won't see any GFP. So if you applied GFP to visualize the amount of autophagy, but you also use baflomycin, you wouldn't actually see any GFP movement. We talk about this one later as well, is necrosis, which is the focus of this section, but we're going over a brief like blurb of all of them. So necrosis is caspase-independent cell death. What this means is that it just fucking happens. Caspases are relatively strict programming molecules in this context, where it goes, if this is here, and this is active, then mark this for apoptosis in this context, which we will get to later. But necrosis is probably a term we've all heard about. You've probably seen really questionable pictures, at least I have, of like the ends of people's toes kind of being all like dead looking. I think I saw a picture of somebody's tongue that was like all white and grayish. Yeah, that's necrosis. Like the tissue is necrosing. Necrotic. Yada yada. Lots of words. There is like a programmed form of necrosis called necroptosis, similar to apoptosis, just necropt. The cell just explodes and dumps everything everywhere, and this can lead to sepsis if it's left long enough, and it can probably kill you. Generally speaking, necrosis leads to an inflammatory response, and cytokines, which are also common for, cytokines are involved in the immune system. They're basically just like, hey, get over here, please. So the cytokine cascade will occur, and it causes additional stress in nearby tissue as well. You see vasodilation, which is widening of your blood vessels, a change in gene expression, and then an increased cell adhesion. The cells will kind of stick to each other more. Now, necrosis is not... Sorry, on my mic. Necrosis is not bad. There are very few things in the body, like natural processes, I mean. There are very few natural processes that are bad. Like, it sounds like, oh, cell death, that sounds bad. There is absolutely a place for this. Especially in the context of like, oh, you have acute injury. You need to have necrosis so that you can bring your immune system over there. Say, like, you cut yourself or whatever. You want to have, like, a little teeny tiny bit of, like, necrosis or inflammation or what have you to signal, hey, immune system, please get the fuck over here and clean this up. So, and, like, plug it up with some, what's the word? I've completely forgot. With some various blood clotting proteins. Or else you just have a cut that wouldn't heal. Kind of a problem. Yes. So it really can be desired in like early stages of infection, but in terms of late stages, you do not want to see this. Especially you don't want to see like long-term necrosis, which is generally kind of bad. But there are many diseases that are due to necrosis, like type 1 diabetes or COPD. I do wonder how type 1 diabetes and necrosis work together. Curiosity. Okay, it's considered a likely mechanism. So in type 1 diabetes, the actual beta cells in your pancreas are dead because it is an autoimmune disease, fun fact, where your immune system just attacks your beta cells, whereas type 2 diabetes is where you just don't respond to insulin the same way. So your pancreas goes, oh shit, the blood sugar isn't going down, I need to make more insulin, and then it overworks and overworks and overworks your pancreas, and your pancreas just kind of like burns out. Like, if you revved an engine really hard and never swapped the gear, you would probably fuck up your car engine. Same thing. So according to National Institutes of Health, which is generally pretty solid, necrotic death considered to be a likely mechanism whereby cytolytic T cells, which are like kill shit cells, including those reactive with diabetes antigens, cause like cell death. So in theory, necrosis could be one of the triggers causing more cytokine, causing more inflammation and more immune cells in where they're not supposed to be, and then they kill off your pancreas. Entirely possible. Now, necrosis can be healed from, like, you don't die if you're necrotic, obviously. It does leave behind a little scar. I don't know if I have any pictures. Let's see if I can find one. There's a scar in the collagen. Ooh, pictures. I'm really glad that I'm doing this shit on a different tab because I can't show you guys most of this stuff. Twitch would get really mad. In the collagen. I would like to see the collagen scar because those are actually kind of fascinating. Oh, that's cringe. Damn. Okay, well, I'm not gonna look that hard. So, there are two major necroptosis. These are the programmed methods. The cell surface receptors, such as TNF-alpha, which is very commonly referred to, it's a tumor necrosis factor. So, TNF-alpha is really great when you don't want to have a tumor, because it makes a tumor die. But it can also be highly upregulated and cause problems, or highly downregulated and cause problems. But the main two things that are really, really important in necrosis necroptosis is RIP1 and RIP3. So, looking over here on the far right. So, this big-ass complex, like, attached to the TNF-alpha receptor, you have RIP1, and if RIP1 kinase, like, pops over here, and then kinases will add a phosphate, so it phosphorylates. So if the RIP1 kinase is active, then the RIP1 and RIP3 will form a nice little complex, and that will signal downstream to cause necroptosis. But if you degrade RIP1 up here, then that's never going to actually happen, or if you have other inhibitory molecules. There's also reactive oxygen species causing injury, which usually targets mitochondria. You see it in, it's ischemia reperfusion injury, which I want to make sure I've got that thing right, because I'm pretty sure I know what it is, but I want to make sure. Could I have it in smaller words? Is defined as the paradoxical exacerbation of cellular dysfunction. So the cell dysfunction is somehow worse and causes more death, following restoration of blood flow to previously tissues that are experiencing stroke or lack of blood flow to the area. So after the blood flow is able to go back, it'll cause more damage somehow, but I'm not going to go in depth on why, but So after a heart attack, just overall you see blood in heart tissue where it really shouldn't be because of that death, and collagen is usually left behind if recovery does occur, as we mentioned with the collagen scars. We don't really know about necrotic cell death in the same way that we do apoptosis. We know tons about apoptosis. Necrosis a lot less so. The receptors do appear to be initiators, and RIP1, which is what I mentioned in here, appears to be a central propagator because so many different things actually do link into it and that signaling pathway. Necrosis does have a physiological role. The assembly of RIP1, RIP3 complexes can regulate programmed necrosis and then virus-induced inflammation, inflammation is really important, as I mentioned before, for immune system responses. So if you want to get rid of a virus, you want to have inflammation so that way your body can go, hey, inflame this area so that way we can get everybody to come here. Because in like an inflamed area of your body, it sounds bad, but it's generally pretty good. Goodbye. Don't interrupt my class! Fucking rude. But you want to have inflammation, because if you can have that inflammation, then the immune system will recognize that and send more immune cells to that area. And if you don't have inflammation, you can't really, like, upregulate the immune response in an area, which is generally kind of problematic if you have, say, a cut, or a virus, or bacterial infection, what have you. You do want to have that inflammation. Pronecrotic complex can be induced in the liver, but if you knock out RIP3 in this context, you can't have tissue necrosis, at least in mice, and it did elevate the viral load and mortality. Viral load is just how much virus you have. But RIP1 itself is kind of the switch between necrosis and apoptosis. You see RIP1 is kind of present here. It just depends on what RIP1 is actually binding to. The difference mainly is that apoptosis involves caspases and necroptosis does not. It's these three pathways, which is either survival, which is NF, I don't know what that is, looks like K to me, but it's not that. So NF activation causes survival. If you have caspases, it causes apoptosis. And if you just have RIP1, RIP3, it's necrosis, necroptosis. So, there are two different things. There is pro-apoptosis and there is anti-apoptosis. There are two different pathways, and what matters a lot in the body, and in this case as well, is the balance between the two of them. There will be certain signals that are trying to upregulate apoptosis and cause cell death, and there are certain signals that are going to try and downregulate it and keep them alive. If you upregulate or deregulate these out of their natural areas, then it can cause problems, which is relatively common in like various pathogens that desire cell death, stuff like that. You will like upregulate apoptosis, and in the case of say cancer will usually try to downregulate apoptosis, so that way it's not eaten or popped out or consumed. So in this context, in very distinctly from necrosis, I always forget to breathe when I'm talking. I'm so bad for that. So in apoptosis, the cells implode, not explode. And they're very neatly removed, so you don't have to deal with like any inflammation in the area. Like you can kind of see like, necrosis, like bleh, just spilling everywhere. But this is an apoptotic cell. There's a bunch of little balls here, which fun fact are legitimately and scientifically called blebbing, which is one of my favorite things. The little white balls here, when you're referring to it blebbing, it's like the ER is generally like these big sheets kind of that fold on top of each other. If you've seen any picture of a cell ever, you know what I'm referring to. But in this case, they form little balls because they're just kind of like popped off and like form little beads because, you know, they're all made of the same stuff, so they're kind of gonna stick together. The same thing with the nuclear envelope here, those little dark balls here, just blobs. But they're pretty neatly removed by the phagocytic cells, which are the ones that eat everything. So apoptosis, you want to think about, how do I distinguish between the three of these, especially in a scientific context, if you're like doing an experiment, being like, okay, what's happening here? Is it apoptosis? Is it necrosis? Is it necroptosis? Is it something else entirely? So there are various different properties, characteristics, that's what I want, characteristics of apoptosis, but there are plenty of them that are shared across the three that I just mentioned. So cells shrink in apoptosis, but they also do shrink during division, so it's not really the greatest indicator, and they do condense, again, similar to in division. But the cell loses contact with its neighbors. The cell junctions between different cells disintegrate, and the junctions and the attachment is really, really important, because if you don't have attachment, you lose the should I divide, should I not signal. The ER, or the endoplasmic reticulum, will kind of dilate and grow and then vesiculates, so it kind of just like breaks down into little vesicles. The cytoskeleton, which is basically just the scaffolding that the cell kind of sits on, will collapse, which does also happen in dividing cells, but a really good indicator is if microtubules are present. Microtubules are really important for pulling apart the chromosomes. They will not be present in apoptotic cells. They will be in dividing cells. The nuclear envelope will break up into those little blebs I was talking about. The chromatin condenses in DNA fragments. The condensing itself is not a very good indicator because that does happen in dividing cells as well. But the cell itself eventually kind of fragments and forms an apoptotic body. This kind of vibe here. But there's no leakage. The main thing is that there's no like I'm dying signal being sent out. The apoptotics are recognized by phagocytes and then consumed. Now, the reason for this is, how do the apoptotic bodies know? Like, how are they being recognized if there's no I'm dying signal? Now, these are because of flipases, which is in fact what they're called. I fucking love this word. Flipases is probably one of my favorite things. So there are multiple different phospholipids. In a cell membrane, you have a phospholipid bilayer is what's referred to pretty much the whole time. When I look down like this, it's because I'm looking down at my hands while I'm trying to like think stuff out loud. There is phospholinositol, and I forget what PS is off the top of my head. But if they're on the outside of the cell membrane, because if there's a bilayer, there's two layers of phospholipids, right? So on the inside, you want to have PI and PS, those kinds of phospholipids. On the outside, you have other phospholipids. And that's pretty normal, and nobody recognizes that as being a problem. But when the flipases can't pull them back in, because they're not naturally going to try and do that. They want to naturally disperse across like pretty evenly, concentration gradient, the whole nine like that. if you can't get them to get pulled back in, then the phagocytes go, that ain't right, and consumes it. Generally speaking, PS is the more important one. High levels on the outer membrane are a really big signal to macrophages or phagocytes. I will use these terms interchangeably. Like, phagocytes are macrophages, but all macrophages are phagocytes. That kind of thing. MFG is a very well-characterized like protein between apoptotic cells and the macrophages, because if this happens, it kind of goes, hey, consume me, and then it will do that. There are two mechanisms of how we make apoptosis happen. There is extrinsic and intrinsic. Now, what this means is that extrinsic comes from the plasma membrane, the outer membrane that I was just talking about, the lipid bilayer, or intrinsic, which is from the mitochondria, which is inside the cell. The mitochondrial damage leads to something called the apoptosome, which is a really fancy thing that we'll be talking about later, but we're going to start with the extrinsic pathway. But the extrinsic pathway does engage the intrinsic. Less so the way, but it does still happen. But while they're independent, they do upregulate one another. And both of them cause caspase activation. However, caspases don't exist in their active form, like, all the time. Because if they exist in their active form, this would happen all the time. They would just do all their shit, right? So, if they're cleaved by other caspases, because that's how you make more active caspases, that one cleaves another one. But if you don't have any active ones to start with, how do you cleave them at all? Now, what happens is that they just kind of like force the initial inactive ones together, and it kind of causes just a small amount of cross-activation in between them, but that's really all you need. It's a very low level of activity, and it causes a conformational change, but one molecule can activate multiple molecules, and then those multiple activate more and more and more and more and more. And outwards, big fancy amplification of a signal until you have like a shit ton of caspases. The regulation of like where the caspases are is key, and that's regulated by adaptor proteins. Like these little guys here will be attached to the caspases. They'll yoink them up, and then they split off, but the actual adaptor proteins will grab them, move them where they need to be, and then they'll get activated. Okay, there are steps to this. Purple is a term that's important to know. So the FAS, we have the ligand and the receptor right here. So the ligands are on the lymphocytes, and the protein little receptors are on the actual target cell itself that will be targeted for apoptosis. So the actual protein binds the receptor. The receptor changes its shape as needed. The adaptor protein, which we just talked about, is the FAS-associated death domain. You will hear this term a lot. It's really metal, to be honest with you. It binds to the cytosolic death domain of FAS. So there's two different death domains. Once they bind together, it goes, okay, we're gonna kill shit. So the death domain recruits the procaspase, which is an inactive form, through another death domain. The complex of the ligand we talked about, the actual receptor, one death domain and procaspase 8 causes this complex called the death-inducing signaling complex. Again, metal as fuck for no reason. Once those caspases, the caspase 8 is cleaved, it will cleave more caspases and onwards. It also cleaves something called BID to TBID, which activates the intrinsic pathway, which we'll talk about later. But this is where you see that activation. Those caspases that are like downstream, they're not the initial caspases, have a couple different degradation targets. They try to break down caspase activating inhibitors, because generally the DNase would, ASE refers to an enzyme. Oh look, Monkey's our favorite. You're late! You are still late to class! I will accept it, because you are paying your tributes. But you are still fucking late. You're late by 30 minutes. Goddamn. So, DNA is like ASE, this little, what is the word? Suffix. This little suffix here will refer to an enzyme, and enzymes break stuff down. So they want to inhibit these enzymes to prevent caspases from, which these enzymes would prevent caspases from chopping up DNA. And you want them to chop up DNA in the case of apoptosis and cell death. Why no go? Just throw a bear at myself. It really should be working. I don't know what the fucking problem is with that one. Oh, is it? Maybe it's because I'm not updated. Why are you silly? Okay. Fine. I'll do it myself. Yeet. Thank you for the yotage. That's very pink. Yeah, the TED software is very pink. Okay, so Rock 1 manages cell junctions and keeps them functional, and as I mentioned earlier, apoptosis results in the degradation of cell junctions. So if you break down Rock 1, you lose the cell junctions. You also see breakdown of a different polymerase, which is the job is DNA repair, which if you're trying to cause death, you don't want the DNA to repair itself. And these different things are involved in the cytoskeleton, and the cytoskeleton degrades, as I mentioned. So if you can degrade this, you can degrade the cytoskeleton. This is a giant fucking mess. The K point to this is, there are different little decoy receptors that you can make to track cell death. The way that you do this is you clone the receptor, which are these little diamonds here, but then you cleave off the death domains, which are these yellow and red lines here. So if you can get rid of the death domain, you don't have death, but you do have the receptor localizing to where it has to be, so therefore you can track where it goes from there. It's like, no cell die, but can be studied. The intrinsic pathway gets activated by the extrinsic pathway as well, but it is initially activated by cell stress. Cell stress refers to, generally speaking, reactive oxygen species, as I mentioned, causing damage to various different things and fucking around. Usually mito damage, likely through something called BAX, bad, or BAC. These all sound the same, because fuck you. They all contain something called a BH3 domain. We will talk about it a little bit in depth later, but generally speaking, there are BH4, I believe it is, and BH3 domains. Let's see if I can find it. BH3. Okay, cool. So, yes, I am right. So, there are BH4 domains and BH3 domains. If you have a BH3 domain in general, it tends to make it apoptotic, but if you have a BH4 domain, it balances itself out and then it doesn't become apoptotic. But if you don't have that domain to balance out BH3, then this is the apoptotic signal. So these three dudes are the please fucking implode signals. Cytochrome C is something that is involved in the electron transport chain inside the mitochondria. It should not be outside of the mitochondria. Ever. So when it is outside the mitochondria, the cell goes, wait, fuck. And it binds and oligomizes, which means, oligo means several, but it's a little bit more than few. But overall, it just, like, puts a bunch of them together of these apoptotic protease factor, or APAF, one, which I don't know if they pop up in this. Yes. These are adapter proteins as well, so they activate cytochrome C release, binds to that, and they go, shit, we gotta get some caspases. Get some caspases together, activate them, and cause that cascade that we talked about earlier. And that cascade causes apoptosis. The apoptosome, which is a fun word, is formed, which I think... Did I not save a picture of that? No, dude, I would have done that. Okay, hold on. I gotta find a picture. So the apoptosome is a goofy-looking motherfucker. Hold on. Okay. This is a great example. This is what the apoptosome looks like. Come back here, bitch. I love Google Docs. Get the fuck back in there. This is the apoptosome. It looks like a wheel. It is a bunch of shit that binds together and overall just kind of signals, please fucking kill me. So, caspase 9, there's a bunch of caspases that are activated by the apoptosome, and then caspases activate other caspases, and down and down and down. But we also see that caspase from the extrinsic pathway, cleaving BID to TBID, which amplifies the pathway and also amplifies mitochondrial damage. And mitochondrial damage is the original factor, so it amplifies the pathway even more by damaging it further. Now, the mitochondria do release things known as SMAC, which is fun, and Diablo, which is also fun. Those are fun little names to remember. They are inhibitors of the anti-apoptosis pathway. So they're inhibition of inhibition. So you just have apoptosis happen. They also release an apoptosis-inducing factor, which is very clear and nice, and I like it. And it acts independently of those caspases that we mentioned earlier. Now, this anti-apoptotic program is generally what's happening in mitochondria. they block apoptosis by these two things called BCL-XL and BCL2, which are not BH3 domain heavy things. And they bind TBID, so that way it can't cause this triggering of apoptosis. And it can increase apoptotic proteins if a cell needs to die. These two just kind of fuck off. But the mitochondria can lose membrane potential. Now, if you're not familiar with what membrane potential is, it's basically the separation of charge across the membrane. You see more negative on the inside and more positive on the outside. I'm pretty sure. I always forget the direction of the membrane the membrane potential. I'm pretty sure it's negative on the inside. Just tell me something with a charge. Yes. Okay, so it is more negative inside, more positive outside. When you have like an action potential, it flips that and then causes depolarization, causes the signal to go on. But anyway, so the mitochondria itself also has a membrane potential, but this potential is caused by the spitting out of hydrogen ions. So it spits out the positive ions and causes the inside to be more negative. And it utilizes this charge separation to power the production of ATP, or our cellular energy. Now, it can lose this membrane potential and completely fuck up the ETC and stall the ATP production. Why would it lose membrane potential? One great example is just holes. Sometimes you can just fucking poke holes in the membrane. The reason this is the mitochondrial permeability transition pore. It's a hole. If you open this, the protons, the hydrogen, will go, let me back in, and we'll go back in because they want to be evened out. The like vast concentration differences, they don't like that. They do not want that. So what they'll do is they go, let me in, and then just kind of like yeet their ass in there, and then completely the membrane potential is just gone. Who is speaking? Oh, it's Bob. I don't know what their name is. I'm calling them Bob. Oh, it's Alfie. That's what they were. Okay. Now, what if you want to live? What a wild concept. Imagine wanting to live. How fucking cringe would that be? So, you see these decoy receptors in anti-apoptosis. It removes the death bit. Another survival mechanism is that C-flip can mimic caspases, which block the death domains from forming. So, inhibitor of apoptosis, or AIP, blocks the function of those initiator and effector caspases, so you just can't fucking get those caspases to go anymore. And if you find any one of these above things, you will find the rest of these things that also prevent apoptosis, due to the fact that they are structurally very similar. Ring fingers are also zinc fingers. They are little zinc-containing bits that can bind the DNA to other things, and it causes the DNA to form like a ring outwards, and that allows different interactions. This itself can target substrates for degradation, and these little ring fingers act as something called E3 ligases. Now, what E3 ligases are is that ubiquitination involves these like 3 ligases that when they're all kind of there, it goes, please fucking kill me. So it can recruit those target proteins to the E2, because there's E1, E2, and E3, for degradation of the proteasome, so it can get rid of these like problematic or apoptotic promoting like proteins. Now, there are a couple conserved domains across a lot of these inhibitor of apoptosis, which are these little red balls, the BIR domain. It's responsible for caspase inhibition. There are 2 ways it does this. You can either competitively bind to it, or non-competitively. If you have taken any degree of high school biology, you will know what these are. Competitive means that it actually interacts with the active site itself. Non-competitive means that it binds somewhere else, but still makes it so that you can't bind to the active site, usually through a shape change. This is that funny little polyubiquitination process. So these ubiquitins here, these extra little funky dudes, are the please fucking kill me signal. But if you don't have the please kill me signal, then no death. Wow. So, generally speaking, the complex here is upregulated for survival. It'll degrade different signals, it'll degrade problematic issues, that way the cell can still stay alive. But if there is a pro-apoptotic signal, it will cause these things to not be a problem anymore, and this will drive the cell death, the apoptosis. Eventually, what can happen is that these anti-apoptotic signals will lose, and the domains will self-ubiquitinate in response to pro-apoptotic stimuli, and that causes the please kill me. All of our cells are actively fucking suicidal. But at the end of the day, it is all balance. There's apoptotic signals that are going at the same time there are apoptotic signals going. Whoever wins the struggle for power causes what happens to the cell. Excuse me. We talked about the extrinsic and intrinsic pathway. Oh, what the fuck? Thank you, Google Docs. Jesus. So you want to target mitochondria externally because they'll release SMAC, which is this apoptotic dude up here, anti-apoptotic dude up here, and that gets degraded. Since the inhibitor of apoptosis isn't there anymore, apoptosis will occur. So this is anti-apoptosis, so if you can stop having this shit happen, then you're there. So if you can target mitochondria externally, you don't have the SMAC release, and then you don't have the binding and degradation, and then apoptosis. So if you can just, like, target mitochondria externally, you don't have this whole fucking thing happen. Here's a summary. Yeah, it's that thing up there. So the BCL2 family, we talked about BCL2 and BCLXL. They can be pro or anti-apoptosis. They will contain 1 to 4 of these BH domains I talked about earlier. So there's BH1, BH2, BH3, BH4. If you don't have that BH4 domain, it's a critical death domain instead. This is the BH3 thing, the critical death, may die. And then it causes apoptosis. Apoptosis? Yeah, I can speak. Apoptosis. If they do have a BH4 domain, it'll balance out that BH3 and it'll become anti-apoptotic. Ow! Though these domains can bind each other and balance each other out, but the levels of, like, if you have X amount of BH3 only and Y amount of BH4, like, how many, if there's more BH4 proteins, then you're gonna have less apoptosis. Why is this word so fucking difficult? Gonna have less apoptosis. So, for example, there's these 3 dudes that are all kind of inhibiting one another. So, BCL2 will inhibit BAX, which is what I talked about earlier, which causes apoptosis, but the BH3 will drive BAX. So, if there's more BH3 than there is of the BCL2 family, like the BH4 domains, if there's more BH3, the BAX can cause initiator caspases to be upregulated and cause apoptosis. If these are inhibited because there's more BH4 domain having stuff, then it inhibits the caspases. But at the end of the day, it's just this big important ratio. Get back up there. We talked about APAF1, that's the... What the fuck is it? Apoptotic protease factor. God, they never actually use like the... It should be apoptotic protease factor. It should be APF, not APAF. Them are some strong words, easy to stumble on. They're easy to stumble on, and also I'm not the best public speaker. I rush too much through my words. I'm trying to slow down, but I just naturally go brrr. Now, in this case, there's this dude right here. Now, the shitty part of this diagram is right here, where I added BCL2 or BCLXL. In theory, if you have like an inactive BCL2 family protein, an apoptotic stimulus is going to activate it, spit out the internal mitochondrial shit, brain go burr, mouth follows. It'll spit out the internal mitochondrial shit, and if it's outside instead of being inside, the cell goes, ah, fuck, I guess I'm going to kill myself, and kind of does that. What was I talking about? So, when you add this here, that is a cell survival signal. It can, like, increase the expression of anti-apoptotic members, and it also does have other roles. It can antagonize those pro-apoptotic factors I was talking about. It can inactivate them directly by heterodimerizing with them. It can keep the mitochondria functional and have the physical integrity preserved. It can close those gates that we were talking about, the mitochondrial permeability pore. That hole, it can close the hole. Can either change membrane potential, which is what BCL2 does, or it can block the actual release of cytochrome C, which is what BCLXL does. They do slightly different things, but they're both important. Now, where they are is the most important thing, because if you have BCLXL, that's great, But what if it's like all the fucking way on the other side of the cell? It's not gonna do shit. If it's on the mitochondria, great. If it's like in the nucleus for some unholy reason, you're fucked. So where it is really, really, really important. So the pro-apoptotic factors are located in the cytosol, which is the liquid inside the cell, or the cytoskeleton, which is like the scaffolding I talked about outside the skull. They will translocate, which is a really fancy word for move, because we gotta be special. They will move to the membranes caught by those apoptotic stimulus you were talking about before. Usually it exposes the C-terminus of a transmembrane domain, and that goes, please kill me. Now, dimerization is antagonized by those anti-apoptotic factors, but like interdimerization of two things causes death regardless of survival signals, so you often have multiple dimers interacting. But those anti-apoptotic factors are in the membranes, and they automatically exist there. They are made there, they stay there. So they don't have to move. So a really strong death response is needed to overcome the no, we gotta live response. Basically, you gotta want to kill yourself really, really, really bad. Because, you know, we've all thought about, like, tossing ourselves in front of traffic. At least I really hope so. I don't hope so. I hope so that way it's not just me. But I hope so that I don't hope so because I don't, like, want anybody to kill themselves. But there's a difference between, like, man, what if I threw myself in front of this bus? And actively, like, throwing yourself in front of a bus. So you have to really, really want to throw yourself in front of a bus to have that happen. You can't just kind of, like, want it as a passing whim. At least in theory. Thirsty. What to do? So that BH3 domain that I was talking about earlier, it's the pro-apoptotic bit. It's buried in the molecule, generally. So they're not going, if they're buried in the molecule, it can't be recognized, and then apoptosis is not going to be triggered. But if it's exposed by that C-terminus being exposed, then it's going to be like, look at me, die. It's like after you get flashed by somebody and then you immediately want to kill yourself. That. BH3 domain is like that one kid in high school who mooned you for some reason. No, I'm not speaking from personal experience. What the fuck, dude? And you just see this man's ass and you think, what the fuck? And then you regret being alive. The BH3 domain is that. Now, there are a bunch of death signals, and they all start with B, to be confusing as fuck. Oddly specific. Yeah. I was having a conversation with a dude next to me, and dude next to me, I will call him Dude A, did not get along with the vast majority of people. I know exactly why he didn't, because he was kind of an asshole. But Dude B was also an asshole, but he was one of the popular assholes for some reason. I couldn't tell you why. He was a hockey dude, which automatically makes you pretty popular in Alberta. In small town Alberta, anyway. So, he liked to harass people all the time. I was one of the people that he used to harass all the time. So, he walked over as I'm talking to this dude, and, like, they hated him for some reason. I don't know why they hated him that much. He was annoying, but not, like, that problematic. And so, I'm talking to him. I'm facing the direction of Dude B. Dude A is facing me as we're having a conversation. Dude B walks next to Dude A, in my line of sight, pulls down his pants, and I get to see his whole white ass. And the problem is that in hindsight, I'm like, damn, he kind of had a fat ass, though. Now, there was nothing, he really didn't have much else going for him, so he had to have something, so good on him for having a fat ass. But also, dude, I don't want to see your ass. Ugh. I sprayed him with cleaning solution once when he wouldn't leave me alone. It was like vinegar and water in a water bottle. He's not gonna die, it's fine. But he was spraying us with it, like me and my friends. So I went up to him, managed to take it from him, and I sprayed him in the face. Anyway. We're not talking about the fact that I was actually bullied in high school and didn't notice until I was like 20 years old. We're not talking about that. We're talking about Bim Bad Bid and Backs, which sounds like a fucking spell. Why is the furniture in my room floating? I don't really want to talk about what all these things do, but they cause death. The BCL ones are the ones that causes survival. Now, Parkinson's. This runs in my family. I might have Parkinson's. I might not have Parkinson's. We'll find out. But Parkin is an E3 ligase, which causes the degradation of different things. And without it, you get Parkinson's. Now, if you don't have a way to get rid of garbage, the garbage builds up, and then you have a problem. So if you are able to get rid of it, great. If you can't get rid of it and Parkin isn't present, then you have this unnecessary shit building up that can cause damage to various other things and fuck with your ability to do everything, including motion. Now, the E3, E2, E1, whatever, the ubiquitin ligases are really tightly regulated because the addition of just one little ubiquitin is enough to say, hey, please fucking kill me. So we kind of want to tightly regulate that one. There are 3 methods that you can use to turn these ligases on, not like that. You can phosphorylate it through a protein kinase, which is this dude right here, the first one. You can have an allosteric transition, which is, allosteric means it's not binding to this active site here, so it binds to something else. Or you can have a protein subunit addition, usually Parkin, but Parkin uses an accessory protein. And there's also ways you can get rid of the signal. You can phosphorylate it. Usually, it's most like glycosylation, which is the addition of glucose. So you post-translationally add this phosphate on there. You can unmask it by removing a protein. Or you can add a little destabilizing N-terminus by pulling a chunk off. And this kind of goes, I'm alive now. So Parkinson's is a selective loss, or just cell death, of dopaminergic neurons in the midbrain. There's a lot of different breakdowns of the brain. I'm not going to talk about it because I don't want to be here for 7 hours. So if you lose dopaminergic neurons are kind of the make-go signal in a lot of cases, and they're also really important in movement. So if you lose them, you can't generate movement or don't have as good control over your movement, which if you are aware of what Parkinson's is, you know what causes that. You have like the shakies and it's really hard for you to like hold a cup because your hands shake so bad. It can be heritable. It can be caused by damage, either way. Now, it can also, you can see the presence of something called Lewy bodies in some cases, not all of them. They are little within the cytoplasm of, they're little neuronal inclusions, so they're extra shit, little extra protein bits that are chilling in your fucking cytosol, which you don't want. But they also contain alpha-synuclein, which we will come back to a lot in this section. But the only thing is that they are not present in autosomal recessive, which means that they are passed down through the not sex DNA, so they're not sex-linked, so you can get it through either men or female. It's not, like, specific there. It's juvenile Parkinsonism, so I assume that what it means is that you get Parkinson's when you're a lot younger than you should. There are a couple different gene products that are involved. So alpha-synuclein is the one that we talk about a lot, and it's really, really important. It is required for the synaptic vessels to be transported. It's not bad on its own. But if you have a couple missense mutations, which just, like, there's like a mutation in this amino acid, and this amino acid is substituted with something else, and this one's also fucked up, then it can cause Parkinson's disease. This should be capitalized, because I thought I said palladium, and I got confused. This can cause the protein to aggregate in these little Lewy bodies, and that can become the Lewy bodies, like I just said. So what happens here is that the monomers of the Park1 don't form right. Now, most proteins are little teeny tiny building blocks, and then they get built up and up and up into these big proteins that are complexes and do complex shit. So if you fuck up the building blocks, like if you put a pound of sugar into a ton of concrete, the concrete doesn't form right. You know, shit like that. So it's the same idea. If you fuck with the monomers over here, then the actual big fibrils are gonna be fucked up. Scroll all the way down. So Parkin, which is this guy up here, the E3 ligase, will break down alpha-synuclein through the ubiquitin, which are these little yellow balls in this image here. When it's mutated, this doesn't work. Glycosylation is the degradation signal for the, hey, please break down this Parkin, and that's attached to the alpha-synuclein, but if you can't break it down, it doesn't really matter how many degradation signals there are. Now, there's also Park2 Parkin, which is the E3 ligase as well. It tends to inhibit cell deaths, which is, if you don't have any of it, then you have too much death, because if you can't inhibit death, you result in death. Logic. Okay, let's skip that part. This bit right here, the accessory protein, is one of the most important things. Important. Really important to activate Parkin and cause the proteasome complex to be able to degrade shit. You need that. There are seemingly 2 pathways from this standpoint, but there's also Park5, which is more pathways. It is required for the recycling of ubiquitin. It doesn't always result in Parkinson's disease. You're more predisposed to it if you don't have this, but it's not really guaranteed, which implies that there is something else that takes care of Park5's function. There's also a couple of these more, which I'm not going to talk about this one specifically, but DJ1 comes up later, so this protects against oxidative stress, and it goes to the mitochondria. But in disease state, it's always in the mitochondria, so it doesn't really do much. Now, pink is localized inside of the mitochondria in the matrix. It can be involved in protection against apoptosis. We will get into it in depth a little bit later. We're halfway through. So why is it selective for dopaminergic neurons? The reason for this is that something called PAEL, or PALE-R, is primarily expressed in dopaminergic neurons. It gets broken down a lot. Well, no, it doesn't get broken down a lot. If you have too much of that shit, it's like, if it's healthy, you would degrade the PALE-R, and congratulations, you're fine. But if it accumulates, you cause stress in the cell, and the cells die. So, generally speaking, if you can't get rid of it because you don't have the functioning ubiquitin proteasome pathway, which is what Parkin causes, if you don't have the Parkin, you don't have the breakdown pathway, then it accumulates, stresses out the cell, and it kills itself. So, accumulation of both PALE-R and alpha-synuclein can result in that selective death of dopaminergic neurons, and there's a couple different treatments for it, but there isn't actually like a cure. You can kind of just slow it down, or you can treat the symptoms, but it doesn't actually do much. However, it also has some fun little side effects, which I noticed are really, really similar to ADHD. If you have too much dopamine, though, which is... yeah, because this upregulates dopamine, which is funny. Includes like sleepiness, hallucinations, leg swelling, obsessions with food, sex, shopping, and gambling. So it makes you crave more dopamine hits, right? Which is very similar to how ADHD works. Because ADHD people tend to seek that dopamine, the dopamine hit. So that's why you see a higher risk of alcoholism or shopping addiction or gambling addiction or just addiction in general. If you don't believe me, I can pull up various studies, or I can call my boyfriend who has ADHD. Now, Alzheimer's is also a funky little thing from cell death. It is caused by the irreversible loss of neurons in general. You'll often hear that Alzheimer's kind of pokes holes in your brain physically breaks down. That is correct. Parkinson's also pokes holes in your brain. You can usually see, I don't know if Twitch will let me show this, but we'll see what happens. Let's look up Alzheimer's brain. Okay. I don't know why I did that. Fuck off. Okay, so this is a brain with Alzheimer's, and this is a brain that's like healthy and normal. You can very clearly see that the brain with Alzheimer's is broken down because you have physically lost those neurons. Now, if I can find one that also has Parkinson's. Now, the actual brain itself looks relatively functional, all things considered. Like, if you looked at the outside structure, it wouldn't really be that big of a problem. God damn it. Copy the image. So, you see the activity is the main difference that you're gonna see across the brain. I don't know what this is. This is probably a CAT scan. What are you? What kind of scan is this? Thank you for just showing me a picture and not telling me where the fuck it comes from. Brain imaging. What kind of image is it, bro? Brain imaging. Okay, hold on. I'm gonna go colorful brain imaging method. What is the colorful brain scan? What is it? It's because the scan usually take. It's a PET scan. Sick, I was right. What is that, a CAT scan or a PET scan? I know not, but it looks thermal. It is a PET scan. So, what is a PET scan? How does PET scan actually work? Positron emission tomography. It's a functional imaging that uses radioactive substances, radio tracers. Oh, so this is where you get the, oops, turn the music off. This is where you get that, the little injections of the radioactive bullshit, I imagine, to then go, where's the activity happening? But overall, you can clearly see like the high red in the highly active area, and the dopamine is going to be probably relatively present there. But let's see. Dopamine in the brain. Dopaminergic neurons in the brain. Where are you primarily located? That makes sense. Okay, so basically, above this little bulb here, I'm gonna zoom in. So this little bulb here, about here-ish, is the tegmentum, and the dopaminergic neurons will then express towards like different parts of the brain from about here. But here is where you see a good chunk of them. So logically speaking, if you see a lot of activity, you're gonna see highest of it in the areas where like most of them are located, right? And you can pretty clearly see that like there's like the higher activity in the outside of the brain, and you can see that the green has degraded on the outside too, not just in that tegmentum area. So that's kind of neat. Let us zoom back out. Like, neat from a scientific perspective, deeply unfortunate for people that have it. Okay, let me close all my tabs. So, Alzheimer's disease, or just AD for short, it is the most common hereditary disorder. It falls under the dementia umbrella, and it's usually involved with acetylcholine, because if you don't have neurons, you can't have the functional acetylcholine movement. Now, it can be caused by these extracellular plaques or amyloid beta peptides, or these things that are called tangles, which are hyperphosphorylated, which means too much phosphorylation. Hyper is too much, hypo is too little. Tau, which will come in later. This is, the tau bit here is how you distinguish it from Parkinson's, but also Parkinson's tends to target pretty much just dopaminergic, whereas Alzheimer's will just break the whole fucking thing. Most Alzheimer's is just kind of happens. There isn't usually like a really strong genetic link. About 5 to 10% of it is genetic. There's about 4 genes. 3 of them are autosomal dominant, and 1 of them is going to be recessive. Now, presenilin is the 2 big ones that you see a lot. So AB42 is amyloid beta 42. Amyloid beta 40 is fine and normal. I believe this means that there's 40 amino acids. But if you add 2 extra, it's mutant and it's bad. The basic structure at the amino acids seems like it seems like relatively fine, especially if you have, say, like a really big protein. Like, oh, you fuck with 1, it's probably fine. Generally speaking, no. It depends on how you fuck with it too. If you add on more to the protein, that is usually pretty significant. If you add on more or take a bunch off, because that can change the physical shape of the protein that is produced. And if you change the shape, that's like, what was it always called? I drilled this into my brain in second year. Form and function are very, very closely related. Function follows form. So if the shape is different, the function can't occur, because the various things are designed for, like, a circle, but if you have a square, it's not gonna fucking work. You know, square peg, round hole. So there are a couple different genes that can be like overrepresented in AD patients. It doesn't always cause it necessarily, at least in the case of this one, or APOE4, which is this guy, the polymorphism. Now, BAP, or beta amyloid precursor protein, it is ubiquitously expressed, and usually it undergoes proteolytic processing and just secretes amyloid peptides out of the cell, because you do need them. They can inhibit serine protease. It can cause the growth of neurites or neuroprotection. It's necessary. The only problem is that when it kind of forms little plaques in the brain where it's not supposed to be, then that causes damage. But if you knock it out, there's no real evidence of vital function. But if you see increased alpha amyloid beta, then it misfolds into those aggregates. Aggregates are just like aggregation, big chunks of protein. So alpha secretase and beta secretase and gamma secretase. These guys are important, and the order is very important. On these amyloid betas, what matters is what order shit is cleaved in. Pause. Hello, welcome in. You're an hour late, sit down. You're an hour late to class. So beta cleaves up here, alpha cleaves in the middle and gamma cleaves at the bottom. So you want to have alpha cleave in the middle, so that you break down this amyloid beta section here, and then you just pop this thing off, and then it can be used, and it's fine. But if you have beta activated first, and this guy, like, fucks off, then you kind of have this problem where alpha won't really activate. Excuse me. And then you end up with these toxic aggregates of amyloid beta. But gamma secretase is also presenilin. Presenilin. I've never been figured out how to fucking pronounce that. Presenilins. I keep wanting to say presnicillins, and I'm like, that's not what it is. Okay. But overall, you see like a toxic effect only with this increased production of the mutant amyloid beta, and that increases misfolding in aggregates. So this is the shape of what presenilins look like. And gamma secretase is a complex that contains it, amongst other things, and it can cause cleavage at 2 different places. At 49, which is fine, or at 40 or 42, which is kind of, we're not really sure about that one. Amyloid beta peptides that are very similar to amyloid beta can form from gamma secretase, so we don't really know how that happens. Kind of problematic. Okay, let's skip through that bit. Now, tau. What the fuck is it? Okay, I didn't write down what the fuck it is because I never had to know. What is tau? What is it? What is a tau in medical terms? Tau is the major microtubule associated protein, or MAP, of a normal mature neuron. Okay, so it is gonna be the bit that attaches neurons to the various scaffolding stuff. According to NIH.gov, tau in Alzheimer's disease and related tauopathies. Tauopathies is a nice word that rolls off the tongue. So the transport tracks are the microtubules, which if you've ever seen that video about like the protein that's like kind of walking through the brain, that's on the microtubules. Most likely. But if you have too much phosphorylation, it dissociates from microtubules and shit can't be processed across. So if you can't transport stuff, that's kind of a problem, and you have a traffic clog. It's like in real life when you have a giant traffic buildup, and then you're like, oh fuck, I can't get home because there's like 16 cars that are piled up in front of me. Many of these like negative isoforms are linked to Parkinson's, and none actually directly cause Alzheimer's, but generally speaking, it's like usually present. But what causes the selective depletion of neurons? We don't know. That's the thing. Alzheimer's disease is one of those things where we're not really sure the vast majority of the time what the fuck's going on. It's likely plaque formation that is causing cell stress. The plaques really only form in specific brain regions, and there might be pro-plaque factors in some brain regions, there might be anti-plaque factors in others, we don't know. But it activates the immune response, which is not good in the brain. You do not want an immune response in your brain. Actually, I'm gonna go on a brief tangent here. If you've ever heard about Naegleria fowleri, or the brain-eating amoeba, the brain-eating amoeba causes the same thing. The brain-eating amoeba does not want to be in your brain. It's not purposely going into your brain. It's in, like, various bodies of water. That's the word I want. Like, bottles? Bibs? Things? Yeah, various bodies of water. And there are some places with, like, signs, like, hey, please don't go in this because you can get Naegleria fowleri. What happens is that you just kind of get fucking unlucky and it gets up your nose. Because the nose is a pretty direct pathway to your brain through the olfactory system. So if a little teeny tiny amoeba gets in there and then goes, well, I need something to eat, so I'm just gonna like follow this area because there's probably food here, and it goes up into your nose and through the olfactory system, and if it doesn't get stopped, because it usually does, if it somehow doesn't get stopped, it makes its way into your brain, then it'll ping your immune system, which goes into your brain to deal with it. However, your immune system is kind of like buckshot. It is not like a perfectly clean little sniper rifle that like gets rid of one thing specifically. There is a fuck ton of collateral damage when the immune system is activated. And that's usually fine in the rest of the body, but not in the brain. Because if you have inflammation and swelling, then you have that kind of intense swelling in between a very teeny tiny area in the brain and the skull that doesn't move. Like, most of the rest of your body can handle the swelling because you're squishy, but the skull is kind of fucking solid. That bitch hollow. Clonk, clonk, clonk. Yeah, that hole is hollow. Yeah, so if you have inflammation there, there's nowhere for the inflammation to go, so it puts pressure on the brain, and then you die. I think there's only like 2 people that have survived an infection, give or take, and it's been in recent years. Anyway. But if you have those plaques, you have the same thing, just slower. Now, there are some promising treatments that are modifying the disease itself, but the problem is that you often see improving in symptoms, but you don't actually solve the disease, or you see more brain inflammation, which isn't great. So, pros and cons. A lot of people are working very hard on this, but the brain is a very complicated piece of kit, so it's going to be particularly difficult for a lot of people to figure this one out. So they're doing their best, but sometimes it's just fucking difficult. There's not a lot you can do about it sometimes, that's not their fault. So, moving on to P53. It is a tumor suppressor, but it kind of acts like the anti-apoptotic BCL family. In most cancers, it is turned off, because if it's suppressing the tumors, as a cancer cell, you don't want that, so it turns it off. It can control the cell cycle, and if you see the word TF a bunch, it's a transcription factor, so it can upregulate or downregulate transcription of DNA. Now, P53 has different function in the cytoplasm. It can have a DNA binding domain that'll interact with those BCL2 proteins. And it can focus on apoptotic induction and inhibition of autophagy. Now, intrinsic necrosis is also involved in P53. We'll talk about that at lecture 7, because we're only at lecture 6. We only have 8 lectures though, so we're making really solid time, actually. So, oh god, I always type 35 instead of 53 because it's just really easy to fuck that up somehow. But if you knock out P53 out of mice, you see the death early of tumors. Like the dominant oncogenes, or oncogenes means genes that can be cancers. You see more tumors and less survival, basically. So in apoptosis, P53 can induce the death, basically, through transcriptional activation of pro-apoptotic genes. It can also directly induce apoptosis by localizing to the mitochondria through those interactions with those funky little BCL proteins we talked about. It is upregulated in response to DNA damage because, again, if the DNA is damaged and the cell is damaged, you want cell death. But it is, like, contextually active at low levels to manage those reactive oxygen species, like we talked about earlier. If you can't repair it, then apoptosis is induced. And the MOMP, which is back up here, mitochondrial outer membrane permeabilization, so the cell opens, and the mitochondrial factors can all be released. Damn, lecture 6 was tiny! So, P53, not 35, Jesus Christ, and MDM2 are 2 oppositional proteins. They keep each other in check, and once P53 can actually activate, it can bind to the membrane, be displaced, and activate the MOMP pathway. So P53, as a tumor suppressor, causes death, generally, so not a big deal with that. But you generally don't want to have too much cell death either. Now, there are a couple different ways that you can look at cells. You can look at it through like an actual light microscope or a fluorescent microscope if you're like visualizing it with your own eyes. Now, the fluorescent microscope and light microscope both blast your fucking sample with radiation a lot of the time. Now, the problem with that is that you can kind of damage it that way, especially like little teeny tiny guys that don't handle light or fluorescence that well. But TIRF is total internal reflection. So the laser doesn't go through it, it kind of goes like gently across the top or across the bottom. So you don't actually damage your sample as much and you can get like decent enough images, but they're not as good as other images are, but it has its place for sure. Now, apoptosis and necroptosis sound really similar. The main difference is that necroptosis involves RIP1 and RIP3 and the necrosome, but it's still programmed compared to necrosis. It participates in parthenogenesis, which is make more pathogens. Genesis makes more. The process by which a disease or disorder develops. Yeah, so it makes the disease or disorder develop. Yeah. It's a very attractive target for the avoidance of unwanted cell death, so if you knock this out in RIP3, you can't clear out the viruses. So, generally speaking, you kind of want to have this as a function. Like, viral infections have been reported to induce necrosis, but it's really too simple, truthfully, so we can't just say that it's just necrosis being induced. Some viruses will encode apoptotic inhibitors, which will shift the balance towards necroptosis instead of apoptosis. So, back to this fucking thing again. So, this trimer, again, is arguably the initiator. Back to complex 1 here. If we have RIP1 surviving, we get necroptosis. If we have RIP1 and caspases, you get apoptosis. If you have neither, you get cell survival. So what happens if you just have necrosis? It means you don't have the program. That is the main difference. Which I believe I made a big-ass table. I don't know where the fuck my table went. There we go. Nice. What the fuck is this? Oh, convenient. Okay. So, necrosis doesn't have caspases involved at all, but necroptosis will have caspases that are inhibiting that RIP3 complex. So if the caspases are present, then this is not going to happen. That's the main gist of it. Back to... It's hard to find all my shit. Yeah, RIP1, RIP3, and Jesus Christ, I wasn't trying to highlight it. And it's induced by those reactive oxygen species. Whereas compared to necroptosis, you see TNF and pathogens mainly causing the initiation compared to reactive oxygen species, but they are still involved. Okay. A bunch of other regulatory components, which I'm not going to talk about too much because honestly, I don't expect that I'm going to have to know them that much in depth. And it's open book, so I'm taking these with me anyway. But how do you, like, get the signal to go and be propagated elsewhere? So RIP1 kinase induces formation of the necrosome, which is this dude here. This is a really great thing here. So if you have RIP1, like, everybody's relatively present, all is well. Hold on. Yeah, so RIP1, just, like, pull that hoe off. You have NFKB. If you have a caspase, you have apoptosis. If you have neither of them, you just have RIP1. RIP3, MLKL, then you have the necrosome and necrosis or necroptosis. Again, it's just programmed versus not programmed, really. So, you form the necrosome, and... So, there are different ways you can actually block the kinase. But generally speaking, necrostatin 1 will target RIP1 kinase. It blocks necrosis, but it doesn't actually target RIP3. So, RIP1 dimerases with RIP3, and that's really the only RIP1 substrate. So it targets RIP1 kinase activity and does block necrosis, but is it necessarily a necrosis inhibitor? I need to write down whether or not it's going to be. So I'm gonna get to that in a second. Actually, I just have my papers. Let me see if I can find where the fuck it went. Lecture 7, propagation. There we go. Okay, is necrostatin 1 truly a necrosis inhibitor? Let's see, what is necrostatin 1 according to NIH? Necrostatin 1. What is it? It's a RIP1 targeted inhibitor of necroptosis, a form of cell death, blah, 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 blah. But why? Why would you argue that it is? Because there's apparently a reason that it isn't. I'm gonna open up my stream on this other thing so I can look at it over here. Just scrolling through a paper really quick. Okay. NEC1. Necrostatins is a group of compounds named for their capability for necroptosis, among which NEC1 has been used to study the contribution of necroptosis and target RIP1 kinase activity in a wide range of pathological cell death events. I am reading Necrostatin 1 and Necroptosis Inhibition, Pathophysiology and Therapeutic Implications from the National Institute of Health. I would argue yes, that it definitely is an inhibitor of necrosis. Because it is preventing that. Do I have a pen anywhere? It is preventing the complex of RIP1 and RIP3, which is the main thing that causes them to go together. So prevents RIP1, RIP3. Which is what? Does it propagate it or does it? Yes. So it propagates necroptosis. And if you don't have RIP1, you can't have this. You can't have the necrosome. Why would it not be an inhibitor, I wonder? It does so across pathways, preventing cell death through apoptosis and necrosis. I don't know. Seems pretty legit. There was a case where you could... Yeah, RIP3 KO mice die because they can't clear out the viruses. So if you can generally inhibit it in the case of necrosis, then you're not going to have necrosis. I don't know. I'm sure there's a reason why you could argue that it's not, but it's probably going to be easier for me if I just argue that it is. I have a thick-ass stack of paper. Okay, so RIP3 targets it for execution of the necrosis, and calling it execution is actually really applicable to what actually fucking happened. Oh, thirsty. So MLKL is the target of RIP3, and when RIP3 activates it, it moves to the plasma and cytoplasmic membranes, where it can change the ion channel activity and lead to necrosis. Now, autophagy, we talked about in the beginning, is the self-eating. Drives to eat oneself. It is hilariously starvation-induced. If you starve the cell, then the cell will consume itself because it goes, well, I can't make anything, so we might as well use the various proteins that make up the cell to feed other things and do other things with them. Now, there's a bunch of different fucking things here. Now, if a survival factor is present and it hits this receptor tyrosine kinase, or an RTK, the RTK is attached to a PI3 kinase. Now, the PI3 kinase will actually block autophagy. It'll phosphorylate this little AKT dude, and it causes survival. So, overall, if you have AKT being active, it turns on P53 and NF, which is the two things we talked about, that will cause survival. Now, it can be induced by inhibiting mTOR, which is this dude up here, which also phosphorylates AKT. So if you can either get rid of the kinase or get rid of mTOR, then you don't have active AKT, and you don't have all the stuff that is going to either drive survival or inhibit death. It is mTOR is like kind of the master switch for everybody. Like, everybody kind of goes in and out. When activated, it inhibits cell death and upregulates cell life things. There can be an alternative. This can kind of act as like the alternative pathway to apoptosis, because apoptosis needs caspases. But if apoptosis is blocked one way or another, autophagy is a really great alternative to it, because you still end up with like just consume. So Bax-Back deficiencies can block apoptosis because those two things upregulate apoptosis. In the case of, say, etoposide, it's a drug that prevents cancer cell growth and is used in cancer treatments, but when it's applied, apoptosis doesn't really happen. So you kind of need to have this happen sometimes, so that way you can just kind of consume the dead shit. But if all the dead shit builds up, like I mentioned about 16 times, it's bad. So if you can have the autophagy, great! So the autophagosomes do still appear when you apply this drug that prevents apoptosis. Now, the autophagosome I talked about from the first page. We are finally going to talk about it again. It is a double membrane bound structure that is pretty much unique to autophagy. There are multiple different ATG genes is what they're called. It's required for formation. You'll see LC3-2 or just ATG8, which is where those ATG genes come from, are these little markers, little stars here. It's a marker for autophagy, and in theory it is non-selective. However, this is a really questionable statement, because the actual contents that are sequestered are targeted to the autophagosome. So it seems like it's non-selective autophagy, but the actual contents that are being consumed are targeted to the autophagosome. So maybe the autophagosome isn't specific, but the stuff is specific to the autophagosome. If that makes sense. So, funky. More pathways, because fuck you. So mTOR, or just TOR, again, whatever, is the master regulator. If you inhibit, it cannot stop autophagy, because mTOR will turn it off, because it turns a KT on. The outer membrane, like, things, or the ATGs, are removed from the sequestration crescent, which is this little dude here. Because if there isn't anything chilling on the outside, it kind of goes, hold the phone, and, like, fucking kills it, because there's something wrong with it. This is important. There are a shit ton of things that can either cause autophagy, which is in green, or it can prevent autophagy, which is in purple. There's a lot of things that are involved here. There's about 16 different pathways. This probably doesn't characterize all of it. So there are a ton of autophagy genes. They're all the ATGs. You have, like, I think up to ATG 29 from what I've seen, and I'm sure there's more. But overall, there's just this kind of complex that forms. Now, TOR is this guy we talked about, mTOR again. So when TOR is activated, it keeps the ATG13 phosphorylated, so it can't bind to the complex, and ATG1 can't be activated. But if you do have it dephosphorylated when you turn off TOR, then everybody can bind, and it can form this complex from the autophagosome and drive autophagy. Now, it does form, like, in a specific order, which is kind of the gist of this shit here, but fuck if it's not complicated. There are two different complexes that are involved in autophagy. The complex 1, this guy here. Complex 2, this guy here. The main difference is just ATG14 or VPS38. That's it. It also depends on where they go. This guy goes to the pre-autophagy thing and the vacuole, whereas this one goes to the vacuole and the endosomes, which are slightly different, but still close enough. So again, what matters is where it is. It can't do the work if it's not close enough to where it's trying to be, right? Now, what if you wanted to mark this? When you are making these kind of pathways, if you wanted to mark it with, say, GFP, that'd be really great. But if you stick it on the wrong end, it can get cleaved off, and then it's not fucking useful. So, you have to stick it to the end terminus of ATG. Because you see this guy right here, where the ATG4 cleaves that hoe? You need to stick it on the other side. Generally speaking, you gotta, like, move that shit out. So, you can, like, have a use for this free GFP. It can be used to determine if ATG4 is active, for example, because if you attach it to C-terminus, but it's still attached to ATG8, that kind of shows that this cleaving is not happening. But if you do see it removed and it's free everywhere, cool, great. Oh, I do have lecture 9. Yeah, I thought we only had 8. Shit. Well, mitophagy is the same thing that we just talked about with mitochondria. That is the main fucking difference. It is mitochondria instead of just everything. So, Parkinson's is that selective loss of dopamine neurons, right? Now, there's DJ1, which is like a sensor for damaged mitochondria, and Pink1 is the mechanism to get to the mitochondria. So, Parkinson's can't clear out the damaged mitochondria, and the cell ends up dying. So, if you can end up clearing out the mitochondria through mitophagy, great! Now, why can't you clear it out? Because you don't have fucking Parkin anymore. Or you don't have Pink anymore. That can also be a problem. Pink will accumulate on the outer membrane, and then it recruits the ubiquitin and Parkin, and it kind of goes, hey, please kill me. The ubiquitin and the Parkin is, again, the please fucking kill me signal. We like those. We like to die. Dying is great. Dying is fun. Now, generally speaking, you want to have, like, this nasty shit getting rid of. But if you don't have them degraded, you just kind of have these fragmented mitochondria, and that's kind of problematic. Now, alpha-synuclein, again, which is involved here, if you prevent the autophagy, then you're going to see cell death. So if you upregulate alpha-synuclein in this context, you will lose the chaperone-mediated autophagy, or the cell eating of the bad shit that's dead. Then you prevent the clearing out of the bad shit, and the cell dies because it's just overwhelmed and stressed out. Mood. That is the entirety of the course. It is 29 pages long. The other half was on birth. It was almost 67 pages long because I was kind of losing my fucking mind. But yeah, I am so pleased that I got through that. Holy shit, I'm thirsty. Oh, I have thirst. I'm gonna fucking die. Now, I know that I rambled for a very concerningly long time. If you have questions about cell stuff, I can try to answer it. I can't promise it. I cannot answer any medical questions because I am not a doctor. None of this shit is medical advice. I can only say, here's what I learned in this course in university. I'm not a doctor. That's it. So if we have questions, feel free to ask said questions. If not, I'm going to switch my light over. Okay. I'm gonna switch my light over and then organize these fucking notes. Because Jesus Christ. Why do I have so many of them? I got way too much shit going on. I do wish that I could have my model and a little hand cam. Like, I'm looking at the possibility of getting a secondary camera set up so I could have, like, a little hand cam. Clunk. Okay. So, we have... starting off strong with necrosis. I should have wrote down on it before I stuck a thing on it. Shit. Okay, so necrosis 1. Off we go. And it's not double-sided because apparently the printer doesn't auto double-sided. You have to do it manually, which is a pain in the ass, so people tend to not do it. Okay. And Pro APOP 2. Pro APOP. Bang. Stick that shit. I also want to keep in mind there's like, here's gonna be the questions. Okay, so death. Decoy. Gotta make sure that I keep those in red so that way I don't accidentally mix them up. So my exam is tomorrow. I have a basketball game tomorrow. Yeah, TLDR. My exam is tomorrow. I'm not that concerned actually. I'm more afraid about the other 2 exams that I have. Like, what do you do, right? I mean, I'm studying, that's what I do. Just study as hard as you can. I'm studying, I'm trying to do all the right things. I'm trying to make sure that I'm getting everything down. Sorry about the mic. Words. Yeah, I'm trying to make sure that I've got everything sorted out as much as I can. So 5, AD. And then just automatic on lecture 6. Wow, look at me go. E53. I think I missed Parkinson's in there somewhere. Necrostatins. This just very quickly becomes like fucking sticky note ASMR. Autophagy. I really wish I had a fucking clip. A little paper clip. That would be really nice right now. And my autophagy. Yeah, I definitely missed Parkinson's. Where did that hoe go? Well, it's somewhere in between lecture 3 and lecture 5. It's the 4th one. 3. Okay, polyubiquitination. That signals Parkinson's. I sound way too excited. Parkinson's. Bing bing bong. Don't go across multiple pages, just be on the one page. Okay. And surprisingly enough, I don't actually have like a functional fucking stapler. I totally thought I owned a stapler. I don't know where that hell went. Okay. The vast majority of it is just the amount of fucking papers I had to have printed off. Okay, so notes. And I'm gonna sort out the extra garbage that I don't have to have. Because at the end of a lot of papers is like the 73 billion references they used. And I can just get rid of these. I don't need to care. Those hoes can't just leave. Okay. So this is the her paper. H-U-R. And this is the Vasepa paper. And I wrote all over these bitches. Because I was like, I am not fucking around today. You're going to have so much stuff. Why do you guys have so many fucking references? You're not supposed to be there. It's not in the right order. 54, 45, 46. So you and you are garbage. You are garbage. Get the fuck out. Oh shit. I have so much paper on my desk. Where did my other paper go? There it is. I'm like, why am I dumb? How did I lose a whole ass paper? How do you do that? A fucking skill. Now, time to see if my hole punch can punch holes in this much paper. Because it may not be able to do so. Hole punching ASMR. I don't think so, seeing as it doesn't want to fit in the hole punch. Joy. I'm gonna try and split it in half. That's a hole! You guys ever read that book in high school? That fucking book that was just called Holes. And it was about as interesting as you would imagine a book about holes to be. I had to read it back to back for some reason. The teachers in my high school, I guess, were just like, you know what we really want to talk about today, kids? Holes. Why did they hate us? I mean, they had plenty of reasons to, but goddamn. The things you do when you don't have access to a stapler. Using fucking rubber bands through a hole. Tie that bitch together. What are you doing? Ta-da! Paper number 1 is all... It looks very cute. It's like a little bow. Okay. Ah! My bands. How am I supposed to go band for band if I don't have any bands? Ugh. You know when you say a sentence and you immediately want to kill yourself? That's how I felt just now. Go in. Why, God? Why am I square peg round holing myself? Oh my God, I probably could fit if I wasn't done. Ow. And you hurt yourself with a fucking hole punch. And it's not like you punched a hole in yourself, it's just that the hole punch is just so hard to punch. Why are you difficult? Yeah. Fight me, bitch. Just really quickly become paper ASMR. Bet you guys thought that your TA was gonna be hot. I mean, the TA is hot. Less papers. I unfortunately need to have all of them. Okay. And I don't think that punching a hole in my notes is the best idea, so I'm just going to tie them together with a rubber band. 5 at a time, you! No! We gonna take them all, bitch. Die. Cringe. Again, you know when you just say something and you're like, actually, why am I alive? It gets stronger. Make me! I mean, I have been trying. My boyfriend and I have been doing, like, actual workouts. When I say my boyfriend and I mean that he has been doing more of the actual workout stuff, and I have been being like, what the fuck are you doing to yourself, peer pressuring you into gym? We don't leave the house. Let me clarify that. There is no leaving the house here. Neither of us want to do that shit. Okay. I've got my notes in order. I'm gonna drop that over there, because that way I won't lose them. It's on top of my tablet, which I need for tomorrow. Bruce Lee got strong with a chair. I mean... We're using, the fucking... What is it called? The little chest that sits at the end of the bed. My boyfriend uses that to do, like, these fucking tricep workouts where he lowers himself down and then back up again. I don't know what it's working. I don't know what it's called, but you'll probably know what it's talking about if I specify. I have 10 minutes. Do y'all want to play Minecraft? Because I do. Butt squats. I think, like, his arms are on the thing, and then he, like, squats in, like, a sitting position, kind of. But it's all his arms. It's not, like, any of his legs. Servers restarted and saved. Thank you, because it started lagging really bad. Okay. Let us get rid of the window capture. And put in... Mine of Craft. Your TA. Doing dumb shit. Meow. Okay, I'm waiting for the game to actually, like, load in. So I'm confident it's there. There you go. Yay! We got blocks. Okay. Now, I've died too many times recently. Let's put this all away. But I finally managed to save up a bunch of shit to get this hoe. Look at her. She's not a hoe. Like, literally exactly not a hoe at all. But I'm gonna be able to get so much fucking deep slate you don't even understand. Y'all don't understand. I'm gonna have so much goddamn deep slate. Stop meowing. Meow. Okay, I have moose, and meese. Moose, and their child meese. Okay. Okay. Let's go back into the whole bees. So this is the tower, which is not coming out as intended, so I think I need to make it about 20 times taller. Yeah, get that slate. Get that slate. Yeah, I think I need to make it taller because it just kind of looks like a weird nub right now. I'm like, this is probably tall enough, right? Wrong. I need more deep slate. And deep slate is a little bit harder to get than actual rock, so it's kind of like, what the fuck, actually? But I'm only gonna be here for the 10 minutes, so I don't really have to change the category. Oh, god, finally. This was so fucking slow earlier, and I'm just like, I'm gonna kill myself. Why does this take so long? It looks nicer, though. It does. Yeah, it looks pretty good, thank you. But I intend to make just a giant fucking tower of evil in the corner somewhere. I want just a tower of death. Tower of death and destruction. I need something else to get. We're gonna use this. Give. Yeah, sometimes the server just lags like that. Like, I broke the block, and then the block is gonna like, oh, actually, we're not broken. I'm like, okay, cool, thank you, server. But that's also just multiplayer Minecraft things. Like, I don't have high expectations in that regard. How much deep sleep do I get? Not enough. Internal circus would be epic. That is the point. That is in fact the goal, but I need so much goddamn deep slate, and I've stolen my deep slate from a couple other people, so it's fine. If need be, I can bring somebody deep slate as an apology, but for now, I'm just stealing everybody's fucking deep slate. And by everybody's, I mean yours, Waki, because that's pretty much all there was. If you need more deep slate, I can bring you more, but I needed it more at the moment. It's my money and I need it now. It's my deep slate and I need it now. There's a big hole. Yay. But here's the thing. So I've just been studying like a fucking mad thing for the last week, and I'm gonna be studying like a mad thing for the rest of this week as well, because my final exams are next Tuesday. So I found a hole. I have a lot of shit to do, and I don't want to do any of it. I've hit the point where I'm just like, I'm so tired. But I'm past the withdrawal deadline, and I really, really don't want to have to retake a course. So I figure that if I just lock the fuck in, I should be okay. And if I fail, will I absolutely want to die? Absolutely! That's not really a new emotion though, so we'll be fine. Which is really fucking morbid, but you know, I gotta make jokes about it. It's funny if you just have been there. Slate of deep. I'm gonna need way more deep slate than I think it's just this. Well, it is a lot of deep slate. Okay, let's get 3 more stacks, I think. I don't think about it. More! Yeah, I'm gonna get 3 stacks. So I'm gonna finish the stack and then get 2 more stacks. Then we'll go from there. Maybe we'll dig. But I probably will not, I'm not gonna stream this the whole time. I'm just gonna hop into Discord afterwards. And I've been thinking about how I really want to use my Discord server more often, but everybody that I know is on the communal Discord server. I'm like, I want to hang out with my friends, but I also want to use my own server. You could extend the mine to bedrock. Oh, I did that earlier! I dug down trying to find some shit, and I hit bedrock really, really fast, actually. Bedrock is a lot closer than you think it is. Not something I really thought I was gonna run into, but lo and behold, I was just like, yo, what the fuck? I haven't seen bedrock since I used to play this as a kid on, like, super flat. What the fuck? Yeah, it was... I'll jump in Discord if I see you in voice. Alright. Well, I'm going to run out the 2-hour timer because I'm really bad at not going all the way to 2 hours. So I want to try and stay at the 2-hour time limit, but I can't study more fucking chemistry. I did my chemistry studying that I was supposed to get done today. I really do have this like little practice quiz that I should be working on, but I'm just like, bro, I'm so tired. I probably should work on it, but I'm just like, fucking Christ. I don't want the tough. I might work on it a little bit later. Because I have got time. Lord knows I've got all the time in the fucking evening, I guess. Not in the world, but definitely all the time in the evening to get this shit done. Okay, don't break that one, because that's where the torch is. Yeah, that's tough. I don't want that shit. Bro! Die. Can't you see I'm fucking busy? I don't have time to entertain you right now. I'm entertaining multiple people at the same time. I'm already being entertaining. At least in theory. Okay, time to get the fuck out because I've got a bunch of deep slate now. Bing, bing, bong. Deep slate is one of those things where you're like, man, I'm probably not gonna need that much of it, and then all of a sudden you realize that you need all of the deep slate. Like, you need that shit. You're like, man. Fuck! So, here we are. I do wish that I could go, like, you know, after you've used the stonecutter, I get why you can't, both logically and for the game purposes, but I wish you could, like, make stuff in the stonecutter and then, like, make it into stuff again. Because the fact that I, like, accidentally made a bunch of stairs once and went, ah, fuck, with, like, a 64 stack. Just made, like, a 64 stack of fucking stairs. Who needs that? Who needs 64 fucking stairs? I mean, I might, but, like, at that moment, I did not. So I just had a lot of fucking deep slate. That was also just a sheep that's here now. Which, fair enough, but also, dude, what are you doing? Armadillos are curled up into a ball. Ugh. Okay. I like the chiseled. It would be between the chiseled and the tiles, but I've kind of already decided on chiseled. Okay. People building a witch tower? I don't remember what the fuck I was talking about, bro. I'm gonna be honest. I forget so fast what I'm talking about. I'm gonna keep that. Think I'm wrong. Okay, so I gotta break that down and build another witch tower. Boing, 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 boing. Oh, people building a witch tower probably need a lot of deep slate, I guess. Probably. It is currently just like a pit of death. You gonna die. But it's not tall enough. Why is there a skeleton somewhere? Damn. Well, time to break all this shit, because what an unfortunate time. It was such a pain in the ass to get, like, put together too, but it's fine. All will be fine once I have my excessively tall witch tower. I don't want to fall down and die! I may be building a witch tower, but I'm doing it on survival and I don't want to fucking die, please. And I got a decent amount of deep slate back from doing this shit, which is kind of nice. I will take that. We're fucking around post-studying, as you do. The amount of times that I've, like, Pomodoro'd myself and just played, like, 10 minutes of Minecraft in between study sessions is truly kind of insane. I'm going to do well on these fucking finals. So help me God, I'm going to do well on these finals. Like, I'm trying so hard. And I think if I don't do well in these finals, that's probably going to be what kills me more than anything, is that I've tried probably harder than I've ever tried on exams in a very long time. Because I have never had to try in finals. Like, not this hard. Like, I've always had to, like, put in effort, sure. But I've never had to, like, lock in and grind. Like, I could have, like, a relatively normal study schedule and be pretty good. And, at least in high school especially, I didn't really have to study that hard. The only reason I studied as hard as I did was so that way I get 99s. Now I study as hard as I do so that way I can get, like, bare minimums. But also that's science for you. Like, but the fact that a lot of science courses are curved, it is a completely different fucking ballgame. Great example is that my, cell course, my intro cell course that I took, like, 2 years ago, had a class average for the midterms we had 2 midterms, and the average for both of them was very close to, 50, like, each time. I think the first one I got a 51 on, and that was a B. Did I study for so long? So you don't get good study habits till much later. Yeah. You will get the good study habits eventually. That's the beauty of it. You will get them. It is kind of funny how it seems like a 64 stack. Ah! Why does it do that? Oh, so 64 stack is 2 perfectly around. I didn't even plan that. Click, 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 click. But oh, anyway, so yeah, so I got a 51 on the first midterm, and that was a B. I got a 70 on the second midterm, and that was like an A+. I think I ended that class with like a high B or low A on average. I like how they have little faces. They really do. That's probably one of my favorite things about them, is they look little dudes. Sup, bitch? Haha, you can't get me, ho. I mean, you could, but I'm far out of your range of perception, so you're not going to get me. Sup, bitch? I saw you. But I'm so far out of everybody's range that nobody can even perceive me. I don't know how I'm going to get down. I think I will just fucking die. Okay. I'll hop off for a second. Okay. Okay. Add to snooze for a couple minutes because that is the 2-hour mark. So I'm going to find somebody to raid because I'm on just chatting. I'm not gonna like swap to something else. This is where we're sticking. Also, shit! Thank you for the raid, Dr. Plague. I was just about to raid out. Thank you for the raid. Thank you so much, actually. And it's really convenient because I'm right here. Perfect convenience. We just joined. So those of you all who are here, go take a lovely little follow to Dr. Plague. They're fun. They're whack as fuck. I appreciate the whack as fuck. You did get to miss the entirety of my cell biology lecture, which is unfortunate. You're late to class. You were so late to class you missed the entire lecture. You were- how? Why are you this late? I saw him at a corpse mountain. Yeah, that sounds about right from- yeah, that seems pretty fucking close to what I'm used to, actually. But, okay, I'll play Minecraft for a wee bit longer, but I'm admittedly not- I'm not doing anything that's interesting. I'm building a tower, so you just get to watch me, like, place a bunch of shit. I may just yap for now. But anyway, to those that do not know me yet for whatever unholy reason, hello, my name is Very Brave Bunny. I'm a variety streamer. I mostly stream Cult of the Lamb and Yakuza, and I will also be doing like Team Up Mondays with my friends, usually, but right now, I am in the middle of finals. So I am doing study with me sessions on Monday for this week and next week, and after that, we're pretty much done. But today, we got to learn all about cell biology, which is funky, fresh. We love that. I love cell bio. It's one of my favorite courses. The midterm/final is tomorrow, which, you know, but what can I do? So I'm trying to study for that one, but it also is open book. But, you know, you do have to know the material if you want to finish it within a normal amount of time. So
Maniacal _ gaming 27 just joined the bunny cult.
Thank you for the follow. Maniacal _ gaming 27. Thank you for the follow, I appreciate it. Yeah, we do goofy shit. We're doing chemistry next week, if that's the kind of unholy shit that you're into for whatever reason. But this Wednesday, I'm gonna be doing Cult of the Lamb, because I'm trying to 100% the game. And I'm hoping, just call me maniacal or mechanical. Oh, I'll shorten it for sure. It's just the TTS going maniacal _ gaming 27 was just the funniest shit to me. But yes, I will shorten it. I'm thirsty, hold on. I've been yapping so much. I'm so thirsty, I'm gonna die. But yes, all y'all are late. You better be on time next class, or else I'm going to lock the door. It's like, or else you're going to be punished by not being allowed in class. I'm just gonna fucking fail your asses, Jesus. I love that's such a funny joke to me. It's like your TA gives you a private lesson, but you're failing. Lock in, bro. Lock the fuck in, dude. Okay. I do feel kind of bad, but I'm also so fucking tired and I really don't want to be on anymore. I've been studying for hours. Yeet! Thank you for the yeet. I am in trouble. Lock the door and I'll use the window. I'll lock the window too, dude. I'll lock all of it. I will lock the whole damn thing. Don't play with me. But also, I got to solve on my model for the whole TA kind of gig, which involved a funky little... Hold on. I want to actually show the whole model, so give me a second. I've got something called brick. I've got something called... I don't know what I've got, but I'm sure I'll figure it out, bro. Okay, hold on. We had like an actual school uniform. I made it like the skirt's a reasonable fucking length, but you know, still fancy. Why is my phone yelling at me? Silence. Okay, let me be big again. Ah. Hello. I am large. There we go. That's close enough. I am a Jeep, but I like the ponytail for today. The ponytail is a vibe. Ignore the fact that I don't actually have this much hair on my main model. It's fine. Don't think about it. It's fucking VTuber magic. Don't stress that hard. But I'm very tired and I feel very bad about rating and then rating. But let's see if we can find somebody to go and bother. Because I enjoy bothering people. Let's see if anybody I know is online. Have I been able to read this person recently? I did. Damn it. I try to like jump everything around a little bit too much. Like I try not to raid the same person like 6 times. Because it usually isn't the best thing on earth, but I don't know. And I'm looking at all these people and I'm like, what is actually happening? Who have I rated? I don't remember anymore. Okay. I've got a vibe. I like this person. This person's cool. I've rated them before. They're fun. Be on your worst behavior. Harassment time. Harassment time is good time. But I will see y'all on Wednesday. Have a lovely evening. Let's go and harass.
A giveaway here and there. Just a few things. Oh, hi, buddy. Hi. Welcome. Thank you for the raid. Wait, Nori. I need you to shout out, buddy. Please. Now. Kidding. Nori's like, you bitch. Thank you. Hi, buddy. Welcome in. What were you streaming today? Mama, you're not a bitch. Shut up. You're a bad bitch. Learning self-bio? That sounds too complicated for my brain. Oh. Is it like a final or... Wait. Is it final season? Is it final season? Thank you, Nori. Oh, okay. What are you majoring if you don't mind me asking? I know, but that's what people say. Mama Fallon is the bad bitch, okay? Gotta put some respect on her name. Okay. Cool. How much longer do you have left? Okay, I think we... Oh wait, where's Joe Entree? Joe Entree. I'm just a little brat. Hey, you're almost there. It's gonna fly by like nothing. I love that for you. I love people in power. This one? This one? You guys, I'm not a brat most of the time. Actually, I am a brat. I'm sorry. Wait. Come closer. Come closer. It's my favorite thing. But how are you other than that, Bunny? Everything going okay? Am I a brat? Wait, Fiery would know this. Fiery. Am I a brat? Tell them the truth. Me. Again? Did you call me Maku? What? You mean Kaku. I'm not a bully. You like it. Fiery likes it when I bully him because it makes him come. Oh, good. Well, I'm glad. I hope you're doing okay. Thank you again for the raid. You're so sweet. Every time that I've gone to like see if I can raid you, I don't think you're online. Because maybe we're in different time zones. Like you stream a little bit earlier than I do. Thank you. Thank you. Oh, okay. So I'm in California. So you are 1 or 2 hours ahead of me. Oh, okay. Yeah. So I do Monday, Wednesday, Friday. I do 6:30 Pacific time until 9:30. So that would kind of make sense. Yeah. Okay. So then you're probably done most of the time by the time I'm done. Yeah. Fiery likes to come. It's not me. He said so himself in the chat. I started off doing 1, and then I started doing 2, and now I'm sticking to 3, only because I also have a full-time job. But eventually the goal is to stream longer and more days out of the week as well. I would really like that, because I like hanging out with you guys, except for Fiery. She's the bully talking shit. I'm not talking shit, I'm talking facts. I like to come. Sometimes. And then I get post-nut clarity, and I'm like, ugh, the fuck was I thinking? Just, being honest. Fiery loves it when I bully him. Clever, shut up. Clever loves me. Wait, is he dead or not? You don't look dead anymore. Oh no, you are dead. Come here, you little bitch. This cocksucker. Bullying is my love language. Honestly, guys, if somebody would say, hey, Kaku, I don't like that, automatically stop. Because I'm just, I'm not about that life. I'm not about making people feel uncomfortable. Except for Fiery, because he loves it. Clever, you don't even reply to me 24/7. Shut up, you liar. Oh, wait, okay, so, Gem, you know the game. I remember Fiery, you too. How do I get him to not be dead again?